This is a production of Cornell University.
Okay. Yeah, so my thesis has largely been devoted to developing computational approaches to characterize plant-specialized metabolism. So if you're perhaps new to this field, one of the first things you might be asking is why characterize plant-specialized metabolism? And in answering this question, I'm also going to find what I mean by a specialized metabolite. So across all of the many thousands of species of plants that exist today, there is a wide variety of morphological traits that is actually mirrored by a very large diversity of chemical traits. Many metabolites are produced across all plants and even more across all organisms. Yet within plants, there are many metabolites that are lineage specific, such as cycazin, which is only made in cycad, Ginkgo B, only found in Ginkgo biloba, and serralin and nimboa are only found in certain monocot or dicot lineages. And so when I say specialized metabolite, I mean a metabolite that is either limited in what plants produce it, or perhaps limited in what organs it is found in, or maybe under what environmental conditions it's produced in. So as you can imagine, across all of the many thousands of species of plants, there's a very, very large number, or estimated total number of plant metabolites, over 1 million. And many of these metabolites have been studied and been found to have useful purposes for humankind, such as pyrethrins, which are potent insecticides, and many specialized metabolites are actually have good medicinal properties, or potent medicines, such as Taxol and artemisinin, and they've been used in for medicinal purposes sometimes since ancient times, and are still used clinically today. So the next question you might have is, well, how are plant metabolites studied? So I'm going to go through a sort of characteristic example. Let's say you take your favorite plant and you believe it has antibiotic properties. So you take an extract of it, you extract metabolites, and you determine through antibiotic assays that this extract actually does have the potential to kill some important bacteria. And so what people do next is figure out what specific metabolites are responsible for this antibiotic property. And so they will use a variety of analytical chemistry techniques to determine the structure of these particular metabolites. And so one of the machines analytical chemistry techniques that is very, very widely used across plant studies is mass spectrometry. And so this is a mass spectrometer shown here. This instrument can detect hundreds to thousands of metabolites in your sample, but it does not identify them. Instead, what it can give you for an individual metabolite is the molecular weight. And this piece of information here, which is called a fragmentation pattern, interchangeably, I'll call it a mass spectrum. And what it is showing you is the masses of substructures in your molecule. And if you know what particular molecule gave rise to a particular fragmentation pattern, you can sort of piece together what the substructures of this molecule is. But if you don't know what molecule gave rise to it, it can be very difficult to determine the compound that is responsible for this fragmentation pattern. But let's say, back to this hypothetical example, let's say you have determined through analytical chemistry techniques the structure of your metabolites that are responsible for the symbiotic properties. And if you want to use this metabolite in any sort of commercial or clinical setting, you'll have to figure out how to synthesize it. And so one of the first things people do is figure out how the plant synthesizes it. And so this involves figuring out what enzymes the plant is using for synthesizing this compound. Often people already know some of the enzymes, and so we'll use co-expression or RNA-seq techniques to figure out or predict the rest of the enzymes in this pathway. And then they validate the enzyme's function by perhaps doing in vitro assays or perhaps transforming plants. And as with in vitro assays, they feed the expected substrate to the enzyme and determine if they get the expected product. And so through this process, the pathway can be elucidated. And what I hope that you're potentially coming away with here is that these two steps can potentially be very time consuming and labor intensive. And so we're considering them to be bottlenecks in the study of plant specialized metabolism. And my thesis is largely going to be involved with trying to alleviate these two bottlenecks. So I'm going to give a little bit more background information on these particular bottlenecks. I'm going to start with the bottleneck of structure elucidation. There are software that exists to take a fragmentation pattern and predict what metabolite gave rise to it. Unfortunately, for largely all of these, they don't typically have good performance on plant metabolite because they are not well trained to recognize plant metabolite. And so they can produce many false positives. One method that is really easy and quick and commonly used is called database search or spectral matching. And in this process, your experimentally generated mass spectra or fragmentation patterns are compared to those in a database. And if enough peaks match or line up, then you can sort of, say, well, at least this molecule here that generated this fragmentation pattern, if it's a good enough match, you can say it is exactly this molecule here that gave this fragmentation pattern, or maybe they're structurally related. However, this technique also does not really have good performance on plant metabolites, largely because in public databases of fragmentation patterns, plant metabolites are poorly represented. And so the last bit of background I want to talk about is on the pathway elucidation bottleneck. And the main culprit here is actually the bottleneck of function prediction. So within plants, at least, gene annotation across plants is primarily based on what the annotated function of the gene is in Arabidopsis, which is this plant here, probably the best studied plant on Earth. However, this is a major problem for specialized metabolism, as these genes are often quickly evolving. So, for example, you might have one gene doing a certain function in Arabidopsis, and because of frequency duplication events in different lineages, there might be maybe a dozen such genes. And so it's difficult to transfer the function to those genes, as they're likely not doing the exact same thing. Furthermore, within Arabidopsis, only about 30% of genes are experimentally annotated. So the majority of these genes that we're transferring function across species, we're not sure what the function is. And an example of what this can lead to. So there are software that, or different algorithms that on a global scale across all plants, predict which genes are acting as which enzymes in specific pathways. However, they aren't, well, for example, for this pathway, there's 500 genes predicted to catalyze this enzymatic step. Likely most of these are incorrectly predicted. So the rest of my talk is going to be devoted to my thesis research, which again is trying to alleviate these different bottlenecks. The first one is trying to alleviate the structure elucidation bottleneck. The objective, we've used machine learning to provide metabolites with structural annotations. The goal of this is to achieve broad and quick annotation of metabolomes with high accuracy. So I do want to give a little bit more background to this before I go into the results. So I have mentioned that within publicly available mass spectral databases, there's only a small fraction of metabolites contained therein relative to the total number of plant metabolites estimated to exist across all plants. And so when people are performing untargeted metabolic studies on plants, often what happens is the majority of the metabolites that you detect go completely unidentified. So an algorithm that we devised to provide an alternative method for metabolite annotation is one that would take a particular query metabolite, and instead of providing the specific identification of such a metabolite, it would instead point out what chemistries are involved in this metabolite. So, for example, this particular metabolite would have an ethylcyanidin, a monosaccharide, and a carboxylic acid together. So the use cases for such an algorithm, although it does not identify the metabolite, it can still be quite useful for uncovering stress-altered routes of metabolism. For us, when we started to create such an algorithm, our goals were to achieve better performance in classification than identification algorithms, and also we particularly wanted good performance across diverse plant metabolites. Okay, so now I'm going to go through the training data sets and the methods that we use to create this algorithm. We use two different data sets. The first one is focusing on lipids only. This repository that we use has over 100,000 total lipids in it. Importantly, these lipids all have their molecular formula, and it also has an in silico, i.e., computationally predicted fragmentation patterns for each lipid within this database, and it contains 30 classes of lipids. And so therefore we wanted our algorithm to classify each lipid into its particular class. The second database that we had is much more chemically diverse. We took about 30,000 total compounds of very diverse chemistries from CABI, which is the Chemical Entities of Biological Interest database. And there are many classes within here. It's a very diverse database. But again, what we wanted to do was train an algorithm to take a metabolite and predict what classes it belongs to. So the first type of algorithm that we trained to classify either lipids or diverse chemicals was based just on molecular formulas. These, every compound has a molecular formula, which is in stark contrast to the relatively few compounds that have publicly available structure information. So in making models or algorithms for classifying based on molecular formulas, we extracted the numbers of common elements within this formula, the ratios between them, and we also gave the model the monoisotopic mass of the compound. And this is the only information that it had for classifying lipids. And again, for this first data set, since it has predicted spectral information for it, we were able to take these spectra and then feed that into the model in order to see if that this additional information improves performance. So first, I'm going to talk about the results on the lipid data set when just using molecular formulas to classify lipids. I'm showing three metrics here of the particular algorithm that we trained. Increasingly, values that are closer to 1 indicate increasingly optimal performance. And each dot is a particular lipid class. So you can see that there are many, actually about half of the classes have really high performance. Yet, unfortunately, there was another large portion of the classes that had low performance. And these low-performing classes always had either a phosphocholine or a phosphoethanolamine head group, and these are structurally similar, which meant that the molecular formulas for these lipids were either very, very similar or indistinguishable. And so we thought that perhaps giving structured information would increase performance. And although it didn't make, every class quite high, it did increase performance for all classes. And since, even when looking at the models using just formula information, we for a lot of lipids, we had very high performance, and this encouraged us to go ahead and see what the performance was on the chemically diverse data set. So first, I'm going to show a breakdown of the different chemistries or different classes that we have in this data set, and how many each class is present. So we have some very large classes, however, these aren't really structurally, they don't give you much information. For example, hydrocarbon derivatives is not very informative for biological molecules. They're most always hydrocarbon derivatives. But the ones that are more informative, such as lignins or alkaloids, aren't very well represented in this database. And so what I'm showing you now is the performance of our model on these different classes. And again, what we wanted to see was all four of these metrics, so all four of these colored dots centered around one. We do see that for the large, not very informative classes, but for the smaller classes, unfortunately, the performance is not really good. And we concluded from this that many classes cannot be distinguished from each other based on a formula alone. So we're next thinking, well, how to proceed. Again, given that for most metabolites of most chemistries, they're not very well represented in public databases. And so fragmentation pattern information is not available for most metabolites. And so we also know that the formula alone cannot really accurately take a given compound and accurately tell, and not accurately classify it into the type of metabolism that it's involved in. So at this point, we were sort of wondering what to do next. And then another algorithm emerged to answer the same question. It's called CANOPUS. It was developed by the Gawker group in Germany. And this is the same lab that developed Sirius. So this algorithm was incorporated into this sort of software suite. And it answers, it is essentially it has more extensive applicability than our models. Further development of our models would have likely required extensive experimental data collection to generate these fragmentation patterns in leadership accounts. Since CANOPUS is quick and easy and free to use, we decided to first test its classification accuracy on some in-house data that we had on, instead of looking at very diverse metabolites, we just said, well, can it classify flavonoids and alkaloids? We found that it could. I don't have the time to go into that today, but we have published this information, and you can go find it on my Google Scholar. So in summary of objective one, the models that we trained in molecular formulas achieve good performance on a broad range of select metabolite classes. However, for most metabolite classes, poor performance was achieved. The addition of spectral information increases performance, yet such data is largely unavailable for plant metabolites. CANOPUS is an exciting alternative, and it presents a wonderful opportunity for the annotation of plant metabolites. So the next part of my talk is, essentially here, now that we have CANOPUS, how are we going to use it? So the second part of my talk is devoted to developing techniques for characterizing and quantifying stress-induced changes in the metabolome. The goal here is to understand how plants alter their metabolism in response to stress. And we think that CANOPUS is really going to help us do that. So annotation yields insights on specialized metabolites. That's what, I'm introducing here. So again, across all of the publicly available spectral databases, there's only about 4,000 compounds or plant compounds represented. And this is much smaller than the estimated total diversity of metabolites across all plants. So in order to address this, to use CANOPUS for annotation, we're using it to provide structural information for a majority of the metabolome. Particularly, we want to uncover stress-perturbed metabolite classes and assist in identification of putative metabolic genes. So I'll get to this part in the third section of my talk. Okay, how are we going to do this? We performed an integrated omics experiment from which transcripts and metabolites were simultaneously gathered from Bacterobium to Stachyon in 17 different tissue treatment conditions. And again, we're doing this firstly to capture and annotate as much of the metabolome as possible. And later on, I'll talk about our second goal, which was to alleviate the pathway elucidation bottleneck. So before I go into the details of the experimental design in this experiment, I want to give some background into the experimental design. Again, the goal here was to induce as many metabolic perturbations as possible while studying agronomically relevant conditions. And I want to point out, firstly, that this experiment was generated by a very large collaborative effort between our lab and the Metabolic lab and the Harrison lab, and also the Joint Genome Institute. So there's a lot of people to thank here, but I'll do that at the end. So we chose to study Brachypodium. This, although it's not a crop, it has close evolutionary relationship to pseudocrops and energy grasses, and it is much easier to study than a lot of these species, being a model organism. And also our collaborators have worked extensively with this species. So the conditions that we chose to study, the first one was copper and heat stress. Both of these conditions negatively impact fertility across many crops. For these stress, we collaborated with the Metabolic lab, experts at studying copper responses in plants. The second condition we chose to study is arbuscular mycorrhizal symbiosis. This is agronomically relevant as well, but for the opposite reason as the copper or heat stress. This condition in which the fungus provides symbiosis, the fungal symbiosis provides the plants with minerals. It has been shown to increase crop yields for different species, and it can contribute to sustainable soil management and health. And we are collaborated with the Harrison lab for this. They are definitely experts in studying symbiosis. So yeah, so here's the experimental design in all its glory. We have the symbiosis experiment here, in which we have studied plants in 3 different conditions: a control, low phosphate, and the sort of combined stress of symbiosis and low phosphate. However, the spores were alleviating, the symbiosis is alleviating the low phosphate condition. So this is fully symbiosis, but for this condition, we do think of it as a low phosphate stress. The second condition, or sort of sub-experiment we devised is, again, looking at copper and heat stress. So we have a low copper, plants that got a low copper stress, plants that got a heat stress, and one set of plants that received combined copper deprivation and heat stress. We thought that this condition, this combined stress, would elicit the largest changes in the metabolome compared to these other two. Our hypothesis wasn't totally met here, which I'll go into a bit later. And then finally, to study different organs, we sampled leaves and cones and spikelets from plants that weren't grown in any stress or condition at all. And again, we're sampling both metabolites and transcripts from these tissues. And this study allows for extensive metabolic perturbations to be detected, and also tracking of condition-induced metabolite classes through CANOPUS. Before I get to the CANOPUS results, I want to talk about how the global metabolomes are impacted in these different conditions. The goal here is to characterize and we'll actually quantify condition-induced changes in the metabolome. The metric that I chose to do this with is the relative difference toxicity index. So this measures the average change in abundance of all metabolites in response to each condition. So what I'm showing you here is each condition with the RDPI. The green plots here are for leaves, and the brown plots are for roots in the different conditions. One of the first takeaways I want to point out is that roots appear more responsive than leaves. So if you compare a root, how much the root metabolome is impacted in a particular condition to the leaf metabolome, the root is always higher. Heat and symbiosis elicit the strongest response, especially in the roots. For symbiosis, this makes a lot of sense since the condition is localized to the roots. Okay, so now before we wanted to use CANOPUS across the whole diverse metabolomes of these plants, we wanted to test how well CANOPUS performs on diverse metabolites. And one of the first things I want to point out, when we use spectral matching to identify the different metabolites that we detected in the study, only about 5% of them were identified. However, CANOPUS annotates roughly 81% of the whole metabolome. So that's a big win right there, I think. So we use multiple methods for assessing CANOPUS's performance across a broad array of different chemistries. The first one that we chose to look at was comparing the predictions between CANOPUS and spectral matching. So for example, if spectral matching takes metabolite X and says this is phosphatidylcholine, does CANOPUS take that same metabolite and say it's a glycerol phospholipid? If so, then we're saying CANOPUS is correct. And if not, then it's not correct. So for about 70% of the molecules that were identified through spectral matching, CANOPUS got about 70% of them correct. And for an extra 10%, it was close. It sort of got to the structural neighborhood, but didn't give it the exact correct classification. So the second method that we chose to use to assess how well CANOPUS is performing is to see if metabolites of similar chemistry have similar annotation. So what we used here is, the first thing we did is we took the fragmentation patterns of our different metabolites and quantified how similar they were to each other, and then mapped them onto this network. For example, if two nodes here, each node representing a particular metabolite, if they're close together, then their spectra are very similar, and we can then say, well, their structure is most likely very similar as well. And so the colors of the different nodes correspond to the classification that CANOPUS has given these different metabolites. So, for example, if you look at this subnetwork here with a lot of nodes closely, very close together, CANOPUS is calling them all lipids. These are all phospholipids. Here's another group of glycerolipids, and then phospholipids here. Another group of glycerolipids. This group up here contains many polypeptides or fumaric acid. This group is potentially the most diverse, but we still think there's similar chemistries outlined in here, amino acids and other aminoids, perhaps small metabolites in here. So on the whole, I mean, there's no definitive quantifications to say how well CANOPUS is doing, but we, on the whole, see similar chemistries having similar annotations. And finally, another method, the third method we used is comparing predictions between CANOPUS and other lipidomic software. So there are other software that, particularly just for lipids, take a particular lipid and classify it into a lipid class. So what we did is we compared, well, does this software call something a glycerophospholipid? If so, does CANOPUS also call it a glycerophospholipid? So we're matching the annotations between these two. And again, we find a metric that's really close to 70% correct. So across all of these, on the whole, we concluded that CANOPUS shows good performance. When it is incorrect, it is annotating based on molecular substructures. So for example, if it could potentially take a glycerophospholipid and call it maybe a fatty acyl or something like that. So it's not altogether wrong. So after that, we did decide to use CANOPUS to determine which metabolites are most impacted under different conditions. So we first used CANOPUS, again, for metabolite annotation. And then we determined which metabolites are up/down accumulated in each condition. So either accumulated or depleted. And then we determined which classes are on average most changed in each condition. And a further sort of metric I used was determining which classes are enriched amongst the up or down accumulated metabolites. This is sort of akin to Gaussian enrichment amongst your, let's say, upregulated genes. So I want to sort of walk you through this figure first before I'll go over the conclusions. So for example, if we take this, this subgraph here, this is the, I'm showing the results here for the roots under the copper deprivation. And each subgraph is showing a particular condition. All the roots are on the bottom, all the leaves are in the top. Each metabolite class shown here represents a class that is highly perturbed under this, on average, highly perturbed under this condition. The ones that are in purple are on average strongly depleted. The ones that are in yellow are on average strongly accumulated. And if a class has an asterisk next to it, it is enriched amongst the strongly perturbed metabolites in this particular condition. So one of the first things that we saw when looking across this whole figure is that lipids appear globally stress responsive. Indeed, if you look at the classes that are enriched in any condition, there's only two classes that were ever enriched. One was sphingolipids in symbiosis root. The other was glycerophospholipids that was actually enriched across several different conditions. And so we're concluding from this that lipids appear globally stress responsive, perhaps because they're altering, performing different membrane dynamics under these different conditions. The other thing that we thought was a little unexpected, sort of interesting, is that whenever flavonoids are showing up as a really perturbed class, they were depleted. So here in copper roots, heat roots, roots of the combined heat copper stress, also symbiosis roots, flavonoids were depleted in these different conditions. We're not really sure what this means, but it was sort of antithetical to their role as an antioxidant, particularly under UV stress. And so one other thing that I wanted to do with CANOPUS is chart metabolite co-accumulation patterns. So I used clustering algorithms combined with CANOPUS to uncover groups of co-regulated metabolites and then annotate the metabolites in these groups. So this particular module here is, I'm showing a few interesting modules to me. This one has metabolites that are in above ground tissues and then are only in roots under symbiosis. And so these might be metabolites that are traveling down to the roots to facilitate symbiosis. We're seeing potentially photosynthetases and alpha fatty acyls, which are known to be important in facilitating symbiosis. This module here is showing metabolites only found in symbiosis roots, potentially they're either fungal derived or potentially the plant is producing them in this particular condition. You see sphingolipids, which are important for changing membrane dynamics under this condition. In this module, this is interesting because there's metabolites found in the leaves and also the spices, but not in the stem. So either they're being produced independently or independently in these different organs, or maybe there's transport that we just didn't capture. And this final module here is showing metabolites that are either excluded from the, completely excluded from the roots grown hydroponically, or only found in roots hydroponically, indicating that there might be growth condition specific metabolites. So one last thing that I want to talk about in this section is a interesting response to the combined copper and heat stress that we observed in the metabolome and the transcriptome. So when I showed the RDPI for the metabolome, what we can see here, I didn't talk about this before, but what we can see is that the combined heat and copper stress in the roots is intermediate between the copper alone and the heat alone. This isn't really the case in the leaves. Arguably, it's at the same level as the heat alone in the leaves, but in the roots, it appears intermediate. I delved into this just a little bit more, looking at heat shock proteins and heat shock factors, which are canonical regulators of the heat response in plants. I charted their expression in the roots under heat versus the roots under the combined heat copper stress. And although there is no significant difference in expression on the whole, we did find that for the majority of heat shock proteins, they are less expressed in this combined stress than in heat alone. So we're not sure if this is a good thing. I mean, it might be a good thing. Perhaps a less disrupted metabolome in transcriptome is indicative of a less stressed plant. Yet, perhaps critical recovery pathways aren't being activated. And since we haven't studied the recovery of these plants after the stress, we can't say which one it is, but it is definitely an interesting phenomenon. Okay, so I'm going to wrap up section 2 now with a summary of some of the key findings. So the RDPI calculation is a promising way to assess a condition's effects on the metabolome in transcriptome. CANOPUS is definitely a useful tool for metabolite annotation. Roots appear generally more responsive than leaves. And finally, the combined heat stress and copper deprivation elicited a weaker metabolomic and transcriptomic response in the roots than the heat stress alone. Okay, so this brings me to the third part of my talk. So previously I mentioned that we wanted to use CANOPUS to alleviate the pathway elucidation bottleneck. And I'm going to talk about methods to do that now, particularly our efforts to use integrative omics analysis to facilitate metabolic pathway elucidation. The goal here is to quickly associate candidate genes with metabolites in an untargeted approach. So, assigning genes to pathways is error prone. So I mentioned in the beginning how traditionally pathway assignment has been targeted, i.e. people are looking for enzymes that they believe to be or are trying to determine if they're involved in a given pathway that they've previously been studying. They probably know some of the enzymes involved. So this is a traditional method, but this targeted approach takes a lot of effort beforehand to determine, existing enzymes in the pathway, existing metabolites. And so this targeted approach can take a long time to complete. However, the untargeted approach, which again, we're looking at this pathway in which genes were predicted to be, or predicted to perform enzymatic reactions in a different pathway. This approach is likely yielding a lot of false positive predictions and incorrect predictions. So there's definitely room for other approaches in this space. And so our approach, the goal here is to develop a pipeline to assign putative metabolic activity to genes using CANOPUS annotation and correlation-based approaches. And particularly, we're using the data that was generated in the experiment that I've described previously. So I'm just going to jump right into this pipeline that I developed for predicting metabolic genes. The first step of it, again, is to take the metabolites that we detected in the experiment I described and use CANOPUS to annotate them. These annotations, given that they're not, full identification, it can't pinpoint exactly which pathway metabolite belongs to. If they were identifications, then you would know what pathway or pathways that particular metabolite is a part of. But CANOPUS annotation can give you sort of a neighborhood of what routes of metabolism a particular metabolite is involved in. And then we also took the transcripts that, from Brachipodium. We annotated them with any putative function that we could. And then we used correlation-based approaches to cluster them. And in doing so, we are mapping these transcripts onto the sort of general routes of metabolism that these metabolites are predicted to be in. And so in doing this, we annotated 12,919 genes in Brachydpodium to a general route of metabolism. So this is, on the one hand, good because we're, annotating a large proportion of the genes in Brachydpodium. However, this is far too many to follow up on. And so what we wanted to do next was sort of prioritize genes for further study. And so we decided to study genes that were potentially important for symbiosis by selecting transcripts with specific upregulation in this condition. And this gave us 213 genes. Further, we wanted to take one more step. We wanted to identify transcripts conserved across species, as these transcripts then likely have a very important function. So we chose to use OrthoFinder to identify transcripts in Brachydpodium that had conserved genes to sorghum and or metacargo. And these conserved genes from a different species, calling orthologs. And further, we chose to look at these two species because they have experiments where people studied symbiosis in the roots. And we can further then select transcripts in Brachydpodium whose orthologs are differently expressed in symbiosis, in metacargo, and or sorghum. And this left us with 66 genes to choose from. But in general, you don't have to apply this pipeline to symbiosis. You can apply it to any sort of stress or condition you wish. And it yields broadly conserved genes with activity and stress response in association with certain metabolite classes. So when we applied it to symbiosis, it uncovered both known and novel symbiosis genes. A few of the known genes that it identified as Brachydpodium 54, a phosphate transporter, and CCD7 and D27, which are enzymes involved in the apocarotenoid biosynthesis pathway, which are important for facilitating symbiosis. We also chose to study one novel gene. This is the GDSL lipase. It had functional conservation in symbiosis with sorghum, clustered metabolites with structural annotations, high-end specific expression in sorghum, symbiosis roots. Importantly, it had no predicted paralogs, and these are genes within Brachydpodium that could potentially be doing, having a redundant function. So since OrthoFinder predicted this gene to have no paralog, we chose it for further study. I'm not going to go into everything that we did, but I'll summarize it here. We used the BPI transformation facility to make CRISPR-Cas9 knockout mutations for this gene. And then I screened and genotyped the progeny for selecting homozygous T1 lines. And then we grew mutant and wild type plants under symbiosis in order to determine if there was any difference in colonization rates between these two genotypes. However, I used quantitative real-time RT-PCR to quantify the expression of fungal marker genes, which directly correlates with how much colonization is happening. And it showed no difference in colonization rates between the mutant and the wild type. And so what we think is the case is that there is actually a functional paralog. After I got to this point, we looked at all of the GDSL lipases within Brachydpodium and charted their expression. And the particular GDSL lipase that had the highest sequence similarity to our gene also had very similar expression. So I guess one take home from this is if you are using OrthoFinder to annotate a large number of genes, you will want to make gene trees to validate its predictions on a case-by-case basis. Okay, so in summary of objective 3, with the addition of evolutionary information, our integrative omics workflow was successful in identifying both known AMS and novel genes. However, further work is needed for validation of the importance of the target genes to symbiosis and determination of their specific metabolic activity. So finally, I want to give just a really brief conclusion and future perspectives on these two different bottlenecks. Just I'm going to talk about structure elucidation. So we showed that CANOPUS had good performance across chemically diverse plant metabolites. I think this could be a game changer. Previously, many studies, when they're looking at how different plants are changing in metabolism in response to a certain condition, these studies have focused on a small yet identifiable portion of the metabolome, potentially ignoring the other parts of the metabolome that might be most affected by this condition. Hopefully, CANOPUS can change this. And for pathway elucidation, CANOPUS is also helpful. Their annotations can narrow down which pathways metabolites can belong to, instead of having no information on what the metabolite is and what sort of metabolism or routes of metabolism it's involved in. Now we can get sort of general information about that. And then, as we did, studying metabolite co-accumulation can putatively assign genes to these pathways. An alternative approach is to, for example, take some of these enzymes that are predicted to be involved in the pathway steps of a particular pathway. And you can use co-expression studies to predict which of these enzymes are co-expressed and then potentially likely to be involved in the pathway, potentially more likely than the enzymes that don't show any predicted or expected expression. Okay, so with that, there's a lot of people that I have to thank. I'm going to start with the people here at Cornell who facilitated this research. First, I'm going to highlight my committee members, especially Gaurav. I don't have an estimate for how much of your time you've spent on just facilitating my growth as a scientist. And I can't really thank you enough for that, but thank you, I'll try. The rest of my committee members, Dr. Sumata Basu, Frank Schroeder, and Jocelyn Rose, are also definitely essential. Kept me from going down large and expensive rabbit holes, and also gave me interesting ideas to think of and explore during my thesis. I also have to thank many members of the Moghe Lab. All of these people have helped me, not only with my research, but they took the time out of their day to answer my questions, and I had a lot of them. So thanks to all of you, and also all of the undergrads that either helped me with my research or had the pleasure of mentoring them. It was really great to do that. I'm really glad I had that experience. The experiment that we conducted was in collaboration with the JGI, and particularly Catherine Louie, Ben Bowen, and Krista Pinacchio really just gave us so much information and also answered my questions. And then members of the Metabolic Lab and the Harrison Lab, I would go up to them, again, ask them lots of questions that they have the patience to answer, but also help me with my research, and everybody was just really nice to me and made my time here so much better. The Harrison Lab, same story. I must have asked Armando Bravo and Ian Jablonski so many, maybe 100 questions each, and they had the patience to guide me through setting up a symbiosis experiment, which I've never done before. I have to thank my funding, the USDA NIFA grant, and also Cornell PA funds. Growth chamber staff for fixing the growth chamber whenever it had unexpected problems. Administrative staff for guiding me through any red tape that I had come across. And again, just everybody here for making my time just immeasurably better. I also have to thank the people that helped me get here. And also, people at Cornell that weren't directly responsible for helping my research, but again, helped my growth as a scientist. I had many professors that I worked with. I IOTA'd for them and learned how to be a better teacher, or took their courses and gained a lot of knowledge from that. So thank you to all of these people. Thank you to my professors at Webster, who not only made me aware that I was interested in science, but also were definitely loud cheerleaders for me, especially Mary Priest, who just never gave up, always spent a lot of her time to push me in the right direction. I'm very grateful to her. I'm also very grateful to the Baxter Lab for letting me into their lab as a complete novice and just mess up everywhere. I had just wonderfully patient mentors, especially Greg and Max. I honestly don't think I'd be here without them. And not only answering all my questions, but showing me that a research environment just could be really awesome. Also other members at the Danforth Center for doing the same thing, being wonderful role models, and just allowing me to be there as, an undergrad. And finally, these are people that just made my life so much better. My friends and all of the Cornell graduate students. I don't have a lot of pictures. I didn't mention this, but I didn't have a smartphone until like May. So I didn't want to get pictures of everybody off of the website, but y'all are here in my heart, even though you're not in my talk. My family, I have to thank them. We're not the best at taking group photos. For, taking me outside of work and having fun with me, but also being forgiving when I didn't come home for Thanksgiving. And especially have to thank my fiance, Kevin, shown here with a dragonfly buddy, for being one of the most supportive people that I have in my life, and also for just getting me through everything. Somewhere where you are. Thank you so much. And now I will take questions.
Oh, sorry. What is your strategy from going from having LCMS data for the structure or the class prediction based on the molecular formula, going from having LCMS data to having a molecular formula for a bunch of them?
In the part of the, let me go back to that slide. We didn't actually do that for the data that I presented here. We had that information already. So we already had, you're referring to the lipid blast database that we had, where we took information from lipids and classified them into their respective classes. The molecular formula and the fragmentation pattern was already provided for us. We have done other experiments where we have taken molecular formula and used other prediction tools to predict the formulas, fragmentation patterns, and use other software to predict the formulas. I can go into that. There's one software that works very well that was MS Finder, and then another software really didn't. Serious 4 didn't work very well. And we could, quantify how well the performance was because these were standards. So we knew what the molecular formula could have been.
This has been a production of Cornell University, on the web at cornell.edu.